This video will serve as a brief introduction to buffers. Now, buffers are solutions that resist changes in pH. We can purchase buffers from a catalog. For example, this solution here will buffer a solution at a pH of 4, meaning that if we add acid or base to this, the buffer that's in here will tend to resist changes in pH. So regardless of things that we add to this solution, it should stay about pH 4. This one buffers at pH 7, and this solution buffers at pH 10. So, like I mentioned before, a buffer is a solution that resists changes in pH. Now, here's how a buffer is made. It's made from a weak acid and the salt of its conjugate base. Now, we need to understand what these things are. A weak acid, you know what that is. That's just one of the acids that's not on our list of 6. And the salt of its conjugate base. We've studied conjugate bases, and we've also studied salts. So let's go through an example. But we need to understand that a buffer is made from these two things: a weak acid and the salt of its conjugate base. So for example, this is carbonic acid. It's a weak acid. This is the salt of its conjugate base, sodium bicarbonate. So let's just review how I got the salt of the conjugate base. So if we mix carbonic acid with water, because carbonic acid is an acid, it will donate a proton, and water will accept that proton. So here's what we have. This is the bicarbonate ion. It's lost or donated its proton, and this water has gained the proton. So this carbonic acid is the acid, and this bicarbonate is the conjugate base. So the salt of this conjugate base would just be like sodium bicarbonate, for example. You just replace one of the acids with a metal, and that's what we've done here. We've taken away one of these hydrogens, and we've replaced it with a metal. So acid, base, and conjugate acid. So this is the conjugate base. This would be the salt of the conjugate base. Just as a side note, the bicarbonate ion is important for buffering blood. That is, helping us to maintain a constant blood pH. Because like we learned in class, any significant change to blood pH significantly alters your body's biochemistry and can be very dangerous. So the bicarbonate ion is something you'll study probably once again in biology when you learn about buffers again. Very important to human life as we know it. So let's study a little bit about how buffers work. The addition of a base is neutralized by the weak acid. So for instance, let's say our buffer solution contains this weak acid right here. The addition of a base, that's right here, well, this base will get neutralized by the weak acid in according to this reaction here. So remember, this is able to give off a hydrogen ion and then neutralize this OH just by making water. Similarly, the addition of an acid is neutralized by the conjugate base. So here's our conjugate base, which would be made from adding the salt of the conjugate base. So in order to make this buffer, once again, we would have added the weak acid and the salt of its conjugate base. And the salt of the conjugate base would split up into just the conjugate base. So if we add acid, that acid will react with this conjugate base as follows. So you know that a base can accept a proton. So it will accept one of these protons here. So if it accepts one of these protons, it means that H3O had to give up a proton, turning it into H2O. So now the H2O has been neutralized. So as a review, a buffer is made from a weak acid and the salt of its conjugate base. And a buffer is a solution that resists changes in pH. So if we have a solution and we add base, that base gets neutralized by the weak acid. And that same solution, if we add acid to it, that acid can get neutralized by the conjugate base. So that buffer solution, whether we add a base to it or an acid to it, it will get neutralized and the pH should stay relatively constant.